brain metastases are secondary cancers that travel from a primary location of cancer, like lung cancer, breast cancer, or melanoma. And these cancers seed the brain even after treatment of the primary cancer and cause a second cancer in the brain. And these cancers are notoriously difficult to detect and to model or to try to develop scientific systems to portray accurately what happens in human patients. And they're also very difficult to treat. And currently, if you have a brain metastasis, the median survivorship is 2 to 5 months. And the most common treatment is palliative radiotherapy. And there are very few targeted therapies for brain metastases. The 3 most common sites of brain metastases are, first of all, from lung cancer, secondly, from breast cancer, and third, from melanoma or skin cancer. And we have chosen to profile lung to brain metastases as they are the most common type of brain metastasis.
And yes, let's get into the details then. The therapeutic targeting at the pre-metastatic stage.
Yes. So I mentioned it's very difficult to track the metastatic process in a human patient, because if you can imagine how many steps are involved in the metastatic cascade, first the cell has to break off from the primary tumor, then it has to invade into the bloodstream, then it has to circulate and overcome all the barriers of actually getting through the circulation, and then the cell has to somehow invade into the secondary site, in this case the brain, and that means these cells have to cross the blood-brain barrier, and then they form what we call micrometastases, tiny little spots, and then finally a full-blown macrometastasis, which produces symptoms and is then detected, and very often surgery is done if possible, and if not, palliative radiation therapy. So you can imagine with all of those different stages, at the human patient level, we're not able to detect most of them. Most of the time patients show up with a full-blown brain metastasis. So I felt it necessary in my lab program to develop an accurate model that recapitulates every stage of the metastatic cascade. So we take cells from patient-derived brain metastases, those patients who have had surgery, and we dissociate those cells and then inject them back into immunocompromised mice. And this is like an assay or a model system where we can read out what the human cells will do in what we call an in vivo environment, or in another mammal. And what happens with these cells is when we inject them into the lung, remember that's their primary site of origin, we can actually track through time every stage as the cells make their way to the brain, because we've labeled the cells with a green fluorescent protein. And with mice who can be sacrificed at different stages of time, you can then detect where are the human cells gone, what are they doing. And so we were able to detect a stage that we were quite excited about. What happened is we injected cells into the mouse lungs, and the majority of mice would die of lung cancer burden, because tumors would form in the lung, before they would have any visible signs of brain metastasis. So that means if we were to do an MRI on the mouse, then the brain would be clean. If we were to even remove the brain of the mouse post-mortem, the brain would look normal. And so we would think, oh, we didn't capture the metastatic stage, we missed it. But remember, we tagged those cells with GFP, green fluorescent protein. So what that means is we can take the entire mouse brain, which to all appearances is normal, and we can dissociate it. And what we found is we could recover a very rare fraction of GFP positive cells. And those maybe only 1% got into the brain, and they were not detectable by any usual method. And so we called this stage the pre-metastatic stage, because this must be the stage in the metastatic cascade where the cells have just crossed the blood-brain barrier, they've just invaded into the brain, but they haven't even yet formed a micrometastasis that would be detectable. And remember, the most important thing about this model is we will never be able to detect that stage in a human patient. And yet, if we know what those original invaders are, if we know the nature of those few cells that first cross into the brain, perhaps we can target them and then block or prevent brain metastases from even forming.
And what is being done to further characterize this invasive percent?
So we've recovered those cells back, and now we've profiled them. So we've looked at their gene expression profile, we understand the genes that they express, and now we can do something called connectivity mapping, which means we can take those gene signatures and input them into a big computer program that tells us which drugs would then be able to target that particular gene signature. And now, of course, we have a nice model, an experimental model, where we can test those drugs. And in fact, we found one drug in our recently published cancer research paper from this year. We found one drug that is actually able to prevent those cells from crossing into the brain. So now when we take those brains back, we treat mice with this drug, which is called apomorphine. If we treat mice with apomorphine, then the percentage of GFP-positive cells that get into the brain after our assay is 0. So this is a proof of principle experiment. We don't know if treating human patients with apomorphine would actually prevent their brain metastases, but theoretically, as a proof of principle, this is a first step to try to understand that if we can profile someone's original lung cancer and then somehow have these predictive biomarkers that tell us maybe this pre-metastatic signature will inform us about whether you're at risk of getting brain metastases or not. And furthermore, if we can profile those cells, we can tell you you should be treated preventatively with this drug so that you'll never get brain metastases. So we're hoping that this model will provide a new therapeutic paradigm where we can start thinking about not treating necessarily the full-blown brain metastasis when the horse is already out of the barn. There's no point in closing the door now, but can we get ahead of the metastasis curve with a preventative therapy?